I'm not sure what is happening with everybody, but hopefully we'll get most people on this call, and then anybody who isn't able to join, at least right now, we will be recording this call so that we can send it out to the StaPH-B members. So that will be our workaround for now, and hopefully by next call we'll be able to get a little bit more of the technical difficulties down. So welcome everybody to July's StaPH-B call. Thank you so much for being here. I am excited to welcome everybody who's able to join to this particular call. You probably have noticed I'm not in the office right now. I am actually out on a training, but luckily I'm getting to spend a little bit of time outside, but I didn't want to miss this call because I think that this is going to be a fun one to be a part of, and I'm excited to welcome a couple of different speakers to this month's call. And so I know that there has been a lot of talk recently about wastewater detection of SARS-CoV-2 in wastewater samples. And so there's been a lot of kind of new initiatives that have been surrounding detection and sequencing of the virus in wastewater and in wastewater facilities and treatment plants. And so I invited a couple of different speakers, some from our kind of state government level, but then also from the news team as well, so they can give a little bit of an update on where they're at with the testing, with sequencing, but also I think that there is kind of a goal, I think, with this call to also kind of engage the rest of the audience and kind of open it up to the wider audience at large to kind of have a little bit more of a discussion back and forth about where other states are at, where everybody is with their wastewater testing, how it's going, whether people have seen any kind of like differences in kind of detection methods, or if they've had any successes with certain methods over others. So I think that there's going to be some kind of good information kind of upfront in the beginning to go over these different initiatives, but then also hopefully there'll be kind of a lot of time at the end for some good discussion, some questions, and hopefully some answers, and maybe some kind of interpersonal dialogue between states, and everybody can kind of update them, the group, on where they're at. So I think to begin with, do we want to start with maybe Rika, if you wanted to maybe start with your section, then we'll move on to the news team, and then we'll open it up for kind of discussion and questions after. Does that make sense? I want to give the news people at least a little bit of time to get on in case people are still having problems.
That sounds great. Can you hear me okay?
Yes. Yes.
Okay, let me pull up my slides. My colleague is helping with that.
Perfect. Yes, we can see your slides.
Okay, thank you. So we are all set. Good evening, everyone. This is Rekha Singh. I'm working as CDC Wastewater Surveillance Manager for the Virginia Department of Health. I will be going for a quick overview of wastewater surveillance program in Virginia. Thank you for having me here. And without further delay, let's get started. Next slide, please. So this is the layout of my presentation. I will be talking about the basics of wastewater-based epidemiology, glimpse of VDH wastewater surveillance team, what are our program goals, and we'll discuss about our well-planned Sentinel monitoring program starting sometimes in August of this year. And finally conclude with a quick info on our upcoming live dashboard. Next slide, please. So let me give you some basics. As you know, the genetic material from the virus that causes COVID-19 can be present in human waste even when individuals have no symptoms. Since people may not have symptoms and it can take several days before infected people start to show symptoms. Sewershed surveillance can provide early awareness of new or worsening outbreaks. So wastewater surveillance could be an additional data source to help direct testing and resources to protect public health. Sewershed monitoring has been used to track the prevalence of opioids and other pharmaceuticals like Zika, SARS, norovirus, and it's been used in long-term surveillance for polio. Next slide, please. So how can we use wastewater data in response decisions? Some early studies have shown that the COVID-19 viral signal can be detected in wastewater several days before the clinical cases are identified. Sewer monitoring can provide an indication of the amount of COVID-19 infection and trends of infection rates within these communities. However, many factors influence human epidemiology. But wastewater surveillance testing is fast and data can be available within a week. Data from sewage will enable communities to measure the scope of the outbreak independent from patient testing or hospital reporting and include data on asymptomatic individuals. It can provide decision support for officials determining the timing and severity of public health interventions to mitigate the overall spread of the disease. It can better anticipate likely impact on hospital capacity in order to inform hospital readiness and the necessity of public health interventions. We can also track the effectiveness of interventions and measure the wind down period of the outbreak. And most importantly, it can provide an early warning for reemergence of the coronavirus. So it is a useful leading indicator of clinical cases in the community. If the clinical data is limited, some of the jurisdictions are using it for the near-term case and hospital utilizations. As you can see from the CDC graphics on right side of the slide, the wastewater surveillance can detect the virus ahead of clinical cases, generally about 6 to 7 days sooner. The higher the directional agreement between case data and wastewater data, the more the trends overlap. So the take-home message is wastewater data trend lead case data trend by at least 6 days. Next slide, please. So this is news, and I think Rory will explain this in detail. This is CDC created a COVID-19 National Wastewater Surveillance System, which is an effort to gather data from wastewater surveillance through a data portal. And news is a new tool that complements existing COVID-19 surveillance system. And it is, we can also call it's a CDC coordinated system for health department led wastewater surveillance for SARS-CoV-2. Next slide, please. So we are a part of news, and the project that VDH is working under is for early adopters of wastewater surveillance. Virginia is one of the only 8 states to receive funding for this effort. We have recently received additional funding to advance wastewater surveillance in Virginia. And next slide, please. And here is our team. We have a great team who is enthusiastic about the topic and dedicated to growing this program in the Commonwealth. Next slide, please. And our team is not complete without our inter and intra-agency collaborators, universities, utilities, laboratories, and local health district partners who are supporting us in this program. Let me take this opportunity to acknowledge them for supporting us to lift this project off the ground, a very important step to protect public health. Next slide, please. So as you can see, VDH is currently involved in 2 active work groups with utility and university partners leading SARS-CoV-2 wastewater surveillance in southwestern Virginia and eastern Virginia. Of particular note in this case is Hampton Roads Sanitation District, or HRSD, because we have initiated the data upload process to CDC News with them. Next slide, please. Here you can see where SARS-CoV-2 surveillance is currently happening in state of Virginia. So far, we can say we have pretty decent geographic coverage. We have not yet assessed population coverage, and we are in that process now. However, the more the merrier in this case. And our goal is to facilitate the transfer of data between utilities currently sampling and news. And we are working towards uploading all the data to Decipher. Next slide, please. Again, a quick reminder that the Virginia surveillance program has 3 goals modeled after the CDC goals. The first being data collection, and our intent is to coordinate with utilities and laboratories to optimize data transfer, develop core standards for data collection and reporting, create training material on these core standards. The second is data analysis and visualizations, and our goals are to conduct patient case data overlays onto SewerShed, provide training to partners on use of data, create visualization that relay relevant data to health officials and the public along with appropriate interpretation, and collaborate with stakeholders to improve data analysis and reporting. Lastly, goal 3 is to develop a community of practice for the purpose of facilitating participation for partners, improving data collection and refining analytical methods, identify and resolve issues around data timeliness, data quality, and barriers to reporting or participating. And finally, to create a channel for policymakers to provide feedback on data visualization. Next slide, please. So where we are in a period of transition now between our initial grants goal and goals established by the next year BP3 money. Ultimately, the core purpose of our team remains the same: establishing data sharing with labs and utilities and running a community of practice for the state. Some localized projects will be winding down over the next few weeks, such as Chesapeake, but a few others will be taking its place, like a project in collaboration with FDA and Department of Correction. The largest new goal we will be accomplishing will be the Sentinel Monitoring, which involves wastewater monitoring for up to 25 selected sites weekly throughout the state. We hope to continue our efforts to use this wastewater data effectively by not just uploading it to CDC Decipher, but to create a database and dashboard specifically for Virginia. So here is our example of data set. HRST really is a pioneer in the state of Virginia for this surveillance, as they started sampling back in March 2020. What we see here is the new clinical cases by data represented by gray bars, SARS-CoV-2 viral load represented by line graph, and the timeline of the governor's executive order at the bottom. Initially, what was observed was a substantial peak around July 2020, but that has been surpassed by the peak following holiday season, especially in January 2021. And we were closely monitoring any peak after 4th of July holiday and in specific areas at Virginia Beach. Next slide, please. So what is our surveillance plan? Our team had submitted a proposal to receive funding to enhance surveillance. I'm happy to share that we have received this funding. It would be available starting August of this year. This proposal has 3 main surveillance components, which are Sentinel Monitoring, Localized Projects, and Variant Surveillance. Next slide, please. And I will be explaining these in the upcoming slides. So as I discussed earlier, influent monitoring at 25 wastewater treatment plants will provide a broad view of the level of infection in community and statewide. This data would provide trends on whether the prevalence of the virus is going up or down in advance of patient case indicators. We have created an application process to verify the utility has the capacity to provide the required fields for news, and the state lab DCLS will take care of the sample analysis moving forward once their lab capacity is established. So we are aiming for start date at the end of the BioBoat sampling, which is 20th of August. And this is contingent upon documentation in place since this funding is announced officially, but we are working on the paperwork. And we are planning to give 2 weeks notice to all the selected sites, and we'll set up a kickoff call. And DCLS is ultimately the sole lab coming online in October to support our efforts. And in the meantime, we are planning to work with one particular lab so that it's easier for utilities to work with. Next slide, please. So this is a list of utilities who have applied for the Sentinel monitoring. We are working with our Office of Epidemiology to finalize sampling sites in different geographic areas to better understand the level of infection in a community and statewide. As I mentioned earlier, we are working with our state public health lab for analysis of the samples. Next slide, please. These are rough outline based on zip codes serviced, and the HRST plants serve 1.5 million people in total. Next slide, please. So you might be very interested in this discussion, wastewater variant tracking. So another component of our statewide surveillance study incorporates variant tracking by submitting a sample of positive samples from Sentinel monitoring sites for further advanced molecular detection. So it's incredibly important that we work with our steering committee and larger committee of practice to select Sentinel monitoring sites to capture the scope of disease prevalence to the best of our ability. These technologies include mutation detection by digital droplet polymerase chain reaction, or DDPCR, for known variants of concern, and amplicon-based sequencing, which include first enriching the abundance of the target, that is amplification, then reading much larger regions of the genetic material, since this technology is not just searching for specific mutations. This can allow for the discovery of new ones. Both approaches have their limitations and advantages, and our hope is that the data provided by both will be complementary. Next slide, please.
Rika, are you
Okay.
still there? Oh, sorry, Rika. It sounds like you're kind of cutting off. We weren't able to hear this slide.
Okay, can you hear me now?
Yes.
Okay, sorry about that. This virtual world is a little different. Where was I? So I think you were not able to hear me after slide 18.
Yeah, we haven't seen this or heard this slide yet.
Okay, so let me get started from this. Our partners at HRST have worked on their mutation detection using ddPCR using BioRed assays. They previously shared results with us from their service area targeting the following mutations. HV underscore 6970 del specific to UK B117, and the other one is specific to Brazil P1 and S, African B1351, and the third one is specific to South African B1351, and the last one detects all three. Next slide, please. So DCLS is developing capacity for detection and genomic characterization of SARS-CoV-2 in wastewater samples. They are doing so using FDA laboratory flexible funding model. Approximately 100 samples will be collected with anticipation of sequencing roughly 50 positive samples for metagenomic submission to the designated NCBI bio project. CDC Epidemiology and Laboratory Capacity Cooperative Agreement, ELC, And with this, they have anticipation of sequencing approximately 150 samples during the budget period of 2021-2022. And next slide, please. And DCLS is procuring instrumentation and testing kits for the BioRad prevalence digital droplet PCR SARS-CoV-2 wastewater quantification assay. And they are working with FDA and VDH to identify wastewater treatment plants that align with 21 forward. Next slide, please. DCLS is also reviewing information for protocols and platforms for SARS-CoV-2 genomic sequencing from wastewater RNA extracts. Currently using Arctic V3 primer set with the Illumina MI sequence for clinical samples. Near-term evaluation planned for the Swift Snap SARS-CoV-2, additional coverage panel for clinical samples, and wastewater RNA extract received from partner laboratories. And I think our DCLS partners are online to give some more details if needed. Next slide, please. And the third part of our study is working on some localized projects. Sewershed monitoring upstream of the influent will be employed to answer specific questions such as status of outbreak, support of for lack of patient testing, etc., over the span of approximately 12 weeks per localized project across the state. This slide illustrates how one can collect data for specific geographical areas by changing the sample collection point depending on the desired outcome. For instance, location 1, the influent to the wastewater treatment plant will provide data for the entire sewer shed, whereas location 4 in the orange focuses on a specific area. This approach is similar to how sites within the sewer shed were selected from the Southwest Workgroup project, targeting zip code with higher case counts. Furthermore, our team has worked on a project with a utility partner which operates an expensive pump station service area, which allowed us to really narrow in on targeted areas by overlaying factors such as vaccination rates, by census tract, and other social determinants of health. This slide is a specific example of localized projects. Next slide, please. So lastly, I would like to say a few words about our dashboard. Our team is developing a public dashboard. We are currently leaning towards using Tableau. In this slide, you'll see more of HRST viral load data mapped out, as well as some very basic trends over time. This isn't our final product, as well as we still need to have several conversations with our steering group and EPI team to discuss appropriate suppression and de-identification standards. Protecting sensitive health data appropriately is a top priority when it comes to public data sharing, and we are trying to be mindful of that. Next slide, please. So this is a zoomed example of that, and these are trends for different plants over time. And we're still working on that. So maybe it will, final product might be a little different than that. We are looking at examples from different states nationally and aggressively working to get the finalized product. So that was all. Thank you. And I'm happy to take any questions.
Thank you so much, Rika. That was a great overview of what's going on here in Virginia. I think that we'll probably save questions maybe till the end. I just want to make sure that we give the other presenters enough time to make sure that they have time to present their information as well. So I think now we'll turn it over to the news team. I think Rory and Shatavia at least are on, and hopefully anybody else who is on the news team who wanted to be on this call was able to make it. So I can turn it over to you guys now.
Yep, thanks, Logan. Yeah, I know Logan brought this up at the beginning, but for those that joined late, at CDC, we don't really get a whole lot of opportunities to interact directly with state and local lab bioinformaticians. And so as much as we're happy to share what our vision is for wastewater variant detection and SARS-CoV-2 mutation detection, this is an opportunity for us to get information for what's been working for your lab. What are you kind of envisioning for the future? What hasn't been working? So as I'm going through the presentation, if anybody has comments, questions, suggestions, just jump in. That's really what we're hoping to get out of this. So I'm going to start sharing now. All right, hopefully everyone can see the presentation screen that's up there now.
Yes.
Okay, great. So I'm Roy Welsh. I'm the laboratory lead for our National Wastewater Surveillance System, which we call NEWS, as you heard in the earlier presentation. We're really appreciative of this opportunity to tell you about our work standing up NEWS and our vision for the future, including SARS-CoV-2 viral mutation and variant detection. But when it comes to viral mutation and variant detection, I think it's really important. I wonder if there's a way to get rid of this. Well, this shouldn't be an issue on most of the slides that I don't have a paragraph long title, but sorry about that little header bar in this presentation mode. But the purpose of this slide is really to identify the problem. So we're up against a pretty extreme challenge when it comes to SARS-CoV-2 viral mutation and variant detection from wastewater. Up until this point, there's been a lot of incredible work that's been done to advance pathogen genomics of SARS-CoV-2 from clinical specimens. And we can generally assume that what's in a nasal swab all came from one point introduction. And there has been some co-infection that's occurred, but it's generally safe to assume that you're dealing with just one SARS-CoV-2 genome there. With wastewater, that's not necessarily true. NEWS is set up for the wastewater treatment facility level. And so we're often dealing with, many, many genomes in a single sample. So these treatment facilities serve populations of hundreds of thousands to millions of individuals. And, on top of that, this sample type is inherently challenging. They're loaded with inhibitors. There's a host of other microbes as well as human host DNA floating around in there. The genomes are sometimes fragmented. And, on top of everything, this has been a system of let 1,000 methods bloom. So the wet lab and the bioinformatic methods for identifying mutations from these particular samples are constantly evolving. So I just wanted to put that out there at the get-go before we go into other things to keep that front and center in people's mind when we go through the other challenges and what we're doing. And because that is such a excessive challenge and burden to overcome, when the National Wastewater Surveillance System was set up, it was set up in mind of detection of SARS-CoV-2 and not really that more complicated ask of variant detection. And so I want to start just by reviewing the science underlying our wastewater surveillance system for COVID. There are 4 advantages that we see for wastewater surveillance. First, we know that 50% of individuals infected by SARS-CoV-2 will shed detectable viral RNA in their feces. Now, this shedding happens very early in an infection, and it happens in both the asymptomatic as well as the symptomatic individuals. It happens in children, it happens in adults. This means that wastewater can provide systematic data that reflects both the total infection burden, unlike the clinical surveillance, which is biased towards those symptomatic or severe infections. Second, wastewater surveillance is not impacted by healthcare seeking behavior. So it doesn't matter whether people go to the doctor or there's issues with clinical testing capacity limitations, wastewater surveillance can still provide a robust measurement of community infection. Third, wastewater testing is efficient. We can think of it as really just a large community pooled sample representing hundreds to thousands, even millions of people, like I stated earlier. And then finally, wastewater surveillance is fast. We can get data in hand in 5 to 7 days after that toilet's been flushed. Compare that with about 2 weeks for the clinical indicators, such as case counts or hospitalization. And Rika mentioned earlier that 6-day leading trend. So given that wastewater tends to be a leading indicator of community trends, NWS was established in September of 2020. So I'll just quickly run through this because it was already presented. But in building the system, we made a very intentional decision that the health departments would be the focus of our engagement with the states, not necessarily the utilities or the laboratories. And this was just to ensure that the data that we're gathering is used for the response. So in this schematic, we start over at the top left, and you can see the overall structure here, where wastewater is generated by the communities. It flows into these wastewater treatment plants. Utilities do the heavy lifting of collecting the samples twice per week often, and they send them to the laboratories for the analysis. And it's the laboratories that do the analysis to determine the concentration of SARS-CoV-2 in each sample. And then the health department submits that data to our National Wastewater Surveillance Data System, which provides, consistent data analysis, visualization, reporting tools, all to support public health interpretation and the use of this data. So participation in NWS has grown quickly, and it continues to grow. This map shows all the jurisdictions using CDC funds to support their wastewater surveillance. There's 36 blue states and local jurisdictions that are currently funded through a total of $223 million through 3 rounds of ELC COVID funding. To begin the transition to a sustainable program, NWS has included in the core ELC cooperative agreement for this year. Jurisdictions will receive a total of $34 million this August, so very soon. And the NWS network has grown incredibly fast thus far, and really kind of unprecedented as far as national surveillance systems go. We've also seen rapid increase in the total number of samples submitted. So we have the graph on the right here, where you can see that the first samples that were submitted were around January of 2021. And then as of June, we had over 10,000 unique samples in the system, and we're on track to double that number shortly. So NWS was primarily set up to focus on community surveillance at these wastewater treatment plants. But the system can also accept reports from the facility level surveillance. And there's great interest in using this approach for facility or building level. Long-term care facilities, prisons, universities, all these are of particular interest due to their tendencies to have these explosive outbreaks that are hard to control. However, the data, as you move away from that wastewater treatment facility towards the endpoint, those building levels, it becomes a lot more noisy. And we have ongoing studies to look at these long-term care facilities, correction facilities, to better understand how the performance of facility level surveillance will go. Universities have had great success implementing wastewater surveillance in the 2020-2021 academic year, and they'll continue to do so this year as we see trends start to increase. There are some challenges for NWS. First, not all homes and facilities are connected to sewer systems. There's about a quarter of US homes that are not on a sewer system. And although most of these are in rural areas, there's a lot of septic, even in some of our cities and suburbs. It's also increasingly common for large facilities like universities, correctional facilities to treat their own wastewater on site. Second, a negative result does not mean that there are no cases in the community, only that it's below the limit of detection for our surveillance approach. So wastewater data cannot be used to clear a community, which will be particularly important to continue to message and emphasize as our case incidences continue to decline. And then as you saw on the jurisdiction map, we still have about 14 states that are not implemented in NWS. For some of those, there's hesitancy to implement something that they worry is just a pilot program and won't be continued after COVID. is better controlled. And we're here to assure that we see this as a long-term project. Implementing news is a big lift for jurisdictions, and they're seeking that insurance that this will be worth the effort. Finally, in order to scale wastewater testing quickly, we had to build the system existing lab capacity. So mostly it's done right now by academic and commercial labs, with a few utilities and public health labs providing this testing. However, the more sustainable long-term approach surveillance model is to move to public and environmental health laboratories. And APHL and the laboratory community of practice are already working on resources to build capacity there. All right, so that was kind of the overview of how news got started, what it's after. Now let's kind of switch gears into the topic of today's talk, which is the detection of mutations and variants. So there's a lot of promise of wastewater sequencing to aid our understanding of SARS-CoV-2 genetics within a community. And it's got many researchers and industries and public health partners actively working to generate sequence data. However, this is a new tool, and the analytics and the use of this data are still under development. We recognized early on that one important aspect of this work will be coordinating the submission of this wastewater sequencing data for disease surveillance purposes. And just to start off this section of the talk, I'm going to use and highlight some of our active projects as real-world examples to demonstrate how we've partnered with NCBI and others to kind of really tackle this important issue of coordinating metadata and sequence data submission. So this was a recent announcement that the first 22 new broad agency award went out just recently. And this has been a really successful model for advancing public health, everything from innovation to building capacity to helping reach these underserved communities. The next announcement will likely be sometime later this year, so do keep an eye out for that. And I brought this up because in this latest round of funding, there was an award specifically targeting wastewater sequencing. So this is the 2021 BAA wastewater project that was awarded to UGA. This project will build and validate viral sequencing platforms to detect SARS-CoV-2 transmission at a community level from wastewater. And they'll analyze the methods for genomic extraction, collection, and analysis in order to provide validated methods that can support our national surveillance efforts. And this is noteworthy because we're at a time as case counts are trending upward across the country. A large proportion of those cases are the emerging Delta variant. And all this is happening right when these universities are preparing to welcome students back. Another active project that's underway is one that was initiated by HHS. This is phase 2 of the commercial wastewater testing, and it was awarded to BioBot. So they've got about 12 weeks sampling hundreds of different wastewater treatment facilities across the country. And that collection and reporting has already begun, and they've also included an arm that's devoted to sequencing the subset of these samples. And that data is now publicly available. So here I've got the bioproject link on the bottom for one of those samples from this HHS contract. And there you can see you can get access to the actual sequencing data. You can also get how that data was created. It was all publicly available. Here you can see it's paired end Illumina data off of the NovaSeq, and they were using the Arctic v3 protocol for generating this data. And they were able to get this in using NCBI's new biosample package that's devoted specifically to wastewater sequencing. Now, this is an exciting step, not only for coordinating sequence data from wastewater surveillance in the US, but also connecting these efforts globally. The biosample package has a field for surveillance program specific identifications. That way that we can link these data to the abundant rich metadata that we have for the National Wastewater Surveillance. And while this was done to support SARS-CoV-2, the attributes were designed specifically to be amenable to other health targets and pathogen genomics in wastewater data. So we're working on a little bit more detailed submission guidance with specific examples for our news partners, as well as others that are active in wastewater sequencing. And hopefully these will minimize any effort and redundant data collection and submission moving forward. So just a quick point here. Again, this is titled SARS-CoV-2 for wastewater, but it's built in mind to include other pathogens. We wanted to make sure we included the phage identifiers and standard nomenclature. We have a real goal of making this interoperable and accessible to a large number of people. There's just a small number of mandatory metadata items, and I just wanted to highlight 2 of them real quick for special considerations. One is that geographic location name. There's certainly some concerns around disclosure, although if you're talking about a treatment facility that services hundreds of thousands of individuals, that should be fairly minimal, but we've seen some states just put the state for that location name, and then wastewater population for that wastewater treatment plant. What's the population that it services? There are some plants that are under 3,000, and for our news partners that are generating this data, 3,000 will be the lower limit, and if it's below that number, they'll either leave it blank or add 3,000 there. So I just wanted to make the group aware of that. This group is pretty well versed in the data structure within NCBI, but I just wanted to bring up this slide from NCBI's help manual to highlight that you can have multiple umbrella projects for your data organization. So if you already have some environmental bio project and an umbrella project that you're under for your organization, you can still link to the National Wastewater Surveillance umbrella project if you like. And here I have the slide linking to that umbrella project. You have additional links for other information about our National Wastewater Surveillance, as well as the access to all the active sequencing projects that are actively going on. Underneath this, we have the BioBot bio project, where you can get access to that actual data, as well as additional links up to the umbrella project to see if there's others that have come in recently. And so that's kind of the end for our rails for the data that we've established. And now let's talk about where we're going in terms of SARS-CoV-2 mutation tracking in wastewater. So since these wastewater samples essentially serve as this pooled community sample, one of the more promising applications has been this variant detection and tracking from wastewater samples. And for all of our advances that we have in the sequencing of clinical cases, there's still some major gaps there for those that are hesitant to seek healthcare, those subclinical infections. And so we see wastewater as a complementary but independent system that can better advise that clinical sequence surveillance. I'm noticing the time, and I'm just going to skip ahead through some things, but I have just a couple slides to really drive home that importance of the challenge of this particular sample site. Here, this is an image from outbreak.info. Each row here is an individual SARS-CoV-2 genome. So we have alpha, delta, gamma, and so on down the line. And if you were to identify a specific mutation in your wastewater sample site, such as one of these, you can only say that mutation is present. You can't really say that you have alpha because it's shared between multiple other genomes. And there's only 5 represented here, but if you're dealing with a wastewater sample type from one of these treatment facilities that service millions of individuals, you could have hundreds of different genomes in that sample. So that's really the challenge that we're up against. And so let's talk about what we mean when we talk about wastewater mutation tracking. So when we're talking about mutation tracking, we're either talking about these PCR approaches, whether it's RT-qPCR or RT digital droplet PCR. This approach can tell you the presence of a mutation in your wastewater sample and potentially the trends of these variants of concern, but this approach cannot tell you whether that mutation is directly associated with a particular genome or the proportion of the clinical burden. And that's because we don't really know with some of these variants that are out there, what that does or how that impacts, viral shedding through the GI system. And the other approach is sequencing. And so whether you're talking about targeted S gene or other marker gene or the tiled amplicon approach, this can tell you the relative abundance and diversity of mutations and potentially identify novel mutations. But this can't tell you the total abundance of mutations in the wastewater or the proportion of those clinical burdens or identify a consensus genome. There's actually an asterisk here because there's always a chance that you, with enough depth of sequencing and some of these long read technologies, you could get a consensus genome out for particular sample sites where you're dealing with a very small population. So we don't want to say not never, just not at this moment. Okay, so the use of wastewater for informing variant circulation is still very much in development. Wastewater has been useful for mutation detection, variant tracking trends, but it's unclear the utility for discovery. This is because tracking mutations associated with variants in wastewater can present several public health interpretation challenges. Now, the fact that the SARS-CoV-2 genome makes up only a small fraction of what's in there, and any nucleic acid enrichment process to improve detection could introduce biases as one of them. Many of the SARS-CoV-2 genomes present in wastewater are likely fragmented and consist of many different mutations that are circulating in a community. Also, targeted mutation and specific to a variant is difficult to assess, and it's dependent on that clinical sequencing database that are often biased. And then if there's any mutations in the primer probe binding regions, you'll have dropout and maybe underreporting there. And then we touched on this earlier, but the fecal shedding rate of SARS-CoV-2 and associated variants is still unknown. And this shedding may impact and by the illness duration, the severity of the variants, as well as the immune response from vaccinations. All these together make the relative abundance of variant infections within a community really challenging at this moment. And so I do want to leave time for questions. So I'm going to breeze through some of these common questions that we get and then go into where we are in this initial phase of evaluating what's out there. So if you're probably a normal individual, you're stressed and you're asked to take on wastewater sequencing and you just want a reliable bioinformatic tool or pipeline to use for this type of data, that's been the most common ask. Now, some of you might be a little bit more resilient and you want to come up with your own and identify one. And that's great, and we'd love to hear from that. But what we're trying to assess is that initial ask. Given that there's a lot of publicly available tools out there, which are the ones that provide reliable analysis of this sequence type? And so here's kind of our scheme for how we're approaching this problem. First we're gathering all those tools and methodologies, and also what are the underlying wet lab techniques that go to build those libraries and generate that data, as well as what are appropriate reference genomes to use. And then we're using these sequence simulators to build these in silico model data sets to really test out these different tools. And then we're also have these active sequencing projects where we get access to real sequence data that we can kind of use as well. And then in the end, what we hope to have is a list of model data sets that are built in silico where we know what was gone into them. They're well documented, they're publicly available for others to use for their own tool that they might have developed, and really to have that mutual understanding of what the different tools are able to do and aren't able to do. So when we're talking to each other. So when we look at the different tools, we have 4 key things that we're considering when we go through. The overall mutation detection sensitivity performance is one. And you can imagine, you have different proportion of a virus. If you're after Delta, you can, lower that total read proportion down and see where it drops off. Also, if you're targeting signature mutations, we want to know, what database resources are best. If your tool relies on one, how easy is it to update that? Another key one is link mutations. So for these amplicons that have multiple identifiable mutations on a single amplicon, can we utilize that to build a little bit more confidence in calling these specific mutations are linked to this particular variant? And that gets into the last one of just that variant determination, which is going to be really a challenging nut to crack. we have this table. It's a little busy here, but it's there to show these are the tools that we're using to generate these in silico data sets at this moment. There might be better ones out there. There might be some in this audience that are actively pursuing, their own tool to generate these types of in silico data sets, and we're definitely interested in hearing about that. There's problems, with building these tools. Some of them, if there's a mismatch, it's going to throw an error. You're not going to get that final output. I'm sorry, I realize I've run out of time, so I'm just going to wrap up with the next steps. Is the in silico evaluation. We got multiple projects underway. There's news partners that are generating data, so there'll be public data available to test out whatever tools are out there. We're working on interagency collaborations to really get some rails on this data so that it's accessible and interoperable in our public repositories such as NCBI, and then whatever analysis pipeline that is eventually out there, NCBI supports it. We have that link to our news decipher dashboard, so we can rapidly get that data into public health. So if you have any ideas, please join us. Please reach out. The email contacts here. And just finally end on, we see this not just for SARS-CoV-2, but we're looking to make this a flexible, interoperable structure that's able to rapidly adapt to whatever is next in terms of our changing public health needs. So with that, I'll just thank you, Gail, for your attendance, and welcome any questions that anyone might have.
Well, thank you so much. I know that we are at time now, just because I think that the kind of technical issues at the beginning bled over, and I really apologize for that. But I will be staying on for a little bit longer, just in case people are able to stick around and do have questions that they want to ask. I know that a lot of people probably have other meetings that they have to get to, so I apologize for that. But this recording will be available to anybody who needs it. And also, yeah, we can make sure that the email addresses of everybody who was on this call will be sent out to everybody, so that if you do have any questions, you are able to get in touch with the presenters today. And I do know, so I guess, let's see, we can open up the floor for anybody who has any questions. Is there anything that anyone has that they would like to ask? Oh, I think that there's a question in the chat. So have you seen any mutations that are not detected in clinical public repositories? If so, what do you make of that?
So from the CDC's end, we have not, but I know some of our state partners that are active in sequencing their samples have mentioned some challenging issues with that, and really without access to that actual viral sequence, it's really tough, like other than go sample some more and try to do some culturing. So yeah, it's definitely a challenge there.
All right, it looks like there are a couple of other questions. So the positivity rate per the number of sample collected.
Is that referring to like your success for sequencing, or just
I think so,
positive for
yeah.
SARS-CoV-2?
I don't know, Byung, if you want to clarify both.
Both. Okay. So, we were just having screaming positives across the board for that second wave that was particularly brutal. But then things were starting to get better, and we started to fall into this area where we're seeing a lot of non-detects, so below that lower limit of detection. And it's all tracks pretty well with what we're seeing in the clinical surveillance that's going on. Now, when it comes to success for sequencing, that is really dependent on your wet lab methods. So what BioBot does in terms of their detection of SARS-CoV-2 might be different than what California or others are using. And so while we have seen pretty good trends from reports from others that will track their CT value across the x-axis and their genome depth of coverage across the y, and you see a really nice trend there where you can see for a particular method, let's say like a 30 cutoff value will get you at least 5x or more coverage. But each sample is unique. There's inhibitors, there's other issues at play, so it's not a perfect trend, but yeah, for most methods, you can probably establish that relationship and then really know if I'm going to get the most bang for my buck, where should that threshold kind of lie for whether or not this is going to be forwarded onto sequencing. And I'm assuming it's going to track with your clinical samples too, that same kind of threshold, that you're not going to send stuff for sequencing if it's not below a certain CT value.
Okay. It looks like Frank asked, what open source pipelines are currently under evaluation?
So there's a couple that are out there. Shatavia, I don't know if you want to address this particular one, but we're looking at ones for not just whole genome sequencing, but individual ones. Missouri has a nice one for the S gene SAMREF, and I'm going to try to pull up my notes so I don't butcher them. But the Zurich group has VPipe. So bear with me one sec.
Sure thing. And yeah, I just want to say great presentation, Rory. Really appreciate it. And if anyone comes across a pipeline that they're having success with, please don't hesitate to reach out and let me know. I would be happy to participate in the evaluation process.
Oh, well, hi, Frank, Shatavia. That's great to hear. Anne, are you on the call? I didn't see her. She's a fellow.
Hi, Shatavia. Yeah, I'm here. I didn't know if I should answer or not. So.
No, go ahead. Yeah, good. I mean, you did the hard work. I just was there for like technical support. So yeah, go ahead and take it.
So we actually started using like some known clinical pipelines that we thought maybe we could use, like that Secret and Irma. And then we realized that that's going to be a little challenging. So what I've been looking at, we did use SAM Refiner, and that's for single amplicon right now, but I believe they're trying to adapt that to the whole genome amplification. And then we also, like Rory said, we were looking at the VPipe, which has been adapted for wastewater. And then basically testing a bunch of standalone tools to try and get that sensitivity and mutation detection. So we're looking at LowFreq and Freebase and trying to put all those together. And a lot of the pipelines out there right now are these compilations of these different tools. And we're trying to figure out which one provides us the most accuracy for an in silico data set. So I hope that answered some of your question.
Yeah, no, that was a great answer. And I see you are receiving some backup in the chat as well. So thank you very much. I appreciate it.
Yeah, I thought the links would probably be better than just listing off the name of tools. All right, great.
All right. Are there any more questions? Oh, it looks like some people posted some other links in the chat there. There was a question earlier about read depth and what percentage of reads are needed to find a variant. And I don't know if you guys have done any sort of like research on that, or if you have a good answer for that. So if you have any kind of data supporting this, are there any like read depths or percentages that you have found of reads needed to find variants? Especially I think with those kind of single site approaches.
Yeah, this is going to be context dependent, and it's pretty tough. Like, so for mutation detection, you know, if you're using digital droplet, that's incredibly sensitive. You can get, you know, a lot better detection than sequencing at those low abundance for specific point mutations. And then for, you know, classifying this as a variant that you've identified in your sample, that's, you know, really tough when you're talking about, you know, hundreds of different viral genomes, different, you know, strains in a particular sample. And that's one that I don't have, you know, a great answer for right now.
That's fair. That is definitely a difficult question.
No, but I mean, I would love to come in and just say, you know, this is what CDC recommends and help alleviate some of that burden. I don't want to stifle any type of innovation and creativity, but this is a field that's actively in development, and all the wet lab and bioinformatic methods are being kind of adjusted and continue to evolve as we go. So yeah, we're up against a really tough challenge here.
Have you found more success with any of the wet lab methods over others? Are there any protocols which have kind of jumped out as kind of ones that you're leaning towards, or anything like that?
I mean, so when you're using the PCR-based approach, we've, you know, not done any kind of deep evaluation ourselves, but we've, you know, certainly heard reports from, you know, those involved in these efforts that the digital droplet does have a lot of key advantages in terms of, you know, inhibitors and the like, getting around some of those problems with just the regular RT-qPCR methods that are out there. Now, for sequencing, I don't have a clear answer. I just know that a lot of people have converged on these tiled amplicon approaches. They don't really see a lot of success on just shotgun, you know, those random hexamer sequencing from wastewater. But you know, there's talks with that latest Spheres presentation, the NEB VARSCIP, the long read. They're actively looking for people to try that trial kit for Oxford Nanopore. So I don't have like this one is better than others, but if others are on the call that are active in this area, please step in and give us like what has and hasn't worked for you.
Yeah, so I was going to ask about if you'd looked at any sort of the metagenomic approaches, but it sounds like those are not as successful.
Yeah, I mean, there's a recent publication that I'll share in the chat, but really just the bang for your buck, you have to devote a lot of time and effort to get enough data from those type of methods. Okay, here's that.
Perfect. All right. Is there any more questions? I know it's a little bit past the hour, and I really appreciate everybody staying on and all of the speakers being available to answer questions past the time allotted to. It was a great presentation, and I really do appreciate you being on the call today. I'll stay on for a minute or two more just in case anybody has any other questions. I don't want to stifle anything. In case anybody has any last minute burning thoughts.
I mean, if there's nothing that's on others' mind, one thing